meeting will be recorded. Good. Okay. So thanks everyone for joining us at the SMB Silent Developmental Subgroup Festival. So today is our third session. So thanks everyone for taking the time to join us. And yeah, I guess we can just start with the introduction of our first speaker. So during the session, please feel free to write in the chat any questions if you want to ask any questions to the speakers, or at the end of each talk, there's going to be like a 5 minutes questions, and you'll be free to either raise your hand or to just turn on your camera and just unmute yourself and ask the question yourself as well. So as you feel more comfortable. So our first speaker today is Ander Movilla Miangolarra, and is a postdoctoral researcher at the John Innes Center in Norwich. Ander's research focuses on the mathematical understanding of cellular memory and how this is established. So today he will talk about his latest results about robust gene expression in yeast. So Ander, we're looking forward for your presentation. So the floor is yours whenever you're ready.
Thank you for the introduction. Oops, let me see if I can. There we are. Okay, so hopefully you can now see my screen.
Yes.
As I was saying, thank you for the introduction and for giving me the opportunity to speak here and to present my results. And well, I wanted to talk about epigenetics in yeast and more precisely about some modeling we've been doing, and essentially how the role of the size of particular genomic regions can affect how these behave. And well, I'll start with the biology and then I'll go easing in some of the mathematics. And since I'm going to be talking about epigenetics, the first thing we need to introduce is the nucleosome. So essentially in eukaryotic cells, the DNA helix is wrapped around these nucleosomes, which are essentially histone octamers. They are composed by eight histones of 4 different types. And in particular, we will focus on one of them today, the H3 histone. And that is because it's thought to be one of the most important ones, depending for certain scenarios, especially for this one. And as I said, around which the DNA wraps, is because the tails of this histone can harbor plenty of post-translational modifications. So here in the picture, you can see on the right, you can see these histones protruding away from the nucleosome and the DNA that is surrounding the nucleosome. And essentially, it's these tails that can harbor these post-translational modifications and that can affect transcription. So essentially, that will control whether the gene is expressed or not. So typically we would think that acetylated post-translational modifications of these histones allow for transcription, and methylation would be of a silencing type of post-translational modification. Now, these post-translational modifications are also highly inheritable. And essentially what means is that a cell that divides, the daughter cells will have similar gene expression profiles to the mother cell. So here, our collaborators, Daniel Saxton and Jasper Rijn, they actually looked at this in yeast, and they were looking at this particular locus in budding yeast that has 2 genes. One of them has been substituted by a GFP protein. So you can see if the cell is expressed because it's essentially fluorescent. And this locus is quite well isolated from its surroundings. And actually, as you can see in the video, when the cells divide, it's very well inherited, the pattern of whether the gene is expressed or not. Cells whose mother cell expressed this gene, they are much more likely to express it. Cells where this gene was repressed, their offspring will also be more likely silenced. But then we also have these interesting transitions in between the silent and the expressed state. They are not very likely, but you can see that it occasionally happens that this mother cell that was silent gives one of their daughter cells suddenly becomes expressed and vice versa. Now, this also happens in fishing yeast. They have a very similar locus that can also be under certain backgrounds, can also be either expressed or silent, and it's also very inherited. And importantly for us, there is plenty of experimental data for the switching rates of these two loci. So that essentially will give us some sort of benchmark for the mathematical models we will develop. And the problem you've got here with the inheritance is that essentially, it's some sort of a serial dilution problem, because if you, so essentially when the cells go through mitosis, DNA is replicated, and then you have two identical copies of the DNA. However, the epigenetic marks, the post-translational modifications of these histones, they are not replicated at least directly in the same fashion as DNA. So essentially, you've got this problem where you now have two, but you only have one set of nucleosomes with the appropriate post-translational modifications. And essentially what happens is that newly synthesized nucleosomes, newly synthesized histones are inserted into these genomic regions. So essentially you are diluting every time the DNA replicates, these epigenetic marks, these post-translational modifications are being diluted. And if this was the case, then essentially any sort of epigenetic memory would only last a few generations. It wouldn't last much more than 2 or 3, because it just would fade away. Now, what happens during the cell cycle is that there are certain enzymes that are in charge of rewriting this memory into the genetic locus, into the particular region of the genome. And essentially the way they do that is by identifying the original epigenetic marks, the diluted ones. They essentially bind to them, and then they spread this epigenetic mark throughout neighboring nucleosomes, as you can see in the cartoon. So essentially these are called reader-writer enzymes, because essentially they read the remaining epigenetic marks and then write them to neighboring nucleosomes. And this is this is how it's thought that essentially daughter cells can recover the epigenetic landscape of the mother cell at the end of the cell cycle. Now, in a more mathematical approach to this, or more mathematical viewpoint, you could think that this is some sort of potential landscape. Actually, in cell biology, they also use this sort of analogies, and they call them Waddington landscapes. But here, in the case that I'll be talking about, you've got essentially, I'm going to consider it two-dimensional. You've got acetylation and methylation in your genomic region of interest, and this landscape has two minima, two basins of attractions, two fixed points that can represent either the expressed state, if it's highly acetylated and low methylation, the silent one, if it's highly methylated and low methylation. And as we were seeing, there could be transitions in between these two states. And these transitions could either be what I call noise-induced. So essentially, because these are biochemical reactions and they are inherently noisy, you could have transitions from one state to the other just due to this biochemical noise. But then you could also have replication-driven transitions, which is what I was talking about before. Essentially, what happens in replication, as I was saying, from a highly, for example, from a highly methylated state, you could, when the cells replicate, essentially you are diluting this epigenetic mark. So essentially you're rolling up the marble, the state of the cell, up this potential landscape. And if replication for any reason pushes far enough, then the cell will actually be able to switch to the other basin of attraction. If it doesn't, if the perturbation due to replication is not that hard, not that strong, then it will go back to its methylation, to its original steady state. And now, well, that was still a bit of hand waving, but if we wanted to actually test these ideas, we would need to build a model for this type of dynamics and actually compare it with experimental data. So essentially, this is what we did. We built a model for a heterochromatin in fishing yeast. That was one of the two loci I was talking about before. And essentially, what happens here is that there are certain genes that are located in this locus that has two regions with plenty of repeats that actually isolate the locus from the rest of the genome quite efficiently. And it has an H that also, it's a region of the genome that allows for and essentially enhances the silencing of the region. Now, this was initially the first attempt to modeling was by Dodd and colleagues in 2007. We actually did a similar model. We adapted it to our research question, but essentially this is also the basics of our model, where nucleosomes can be in 3 different states. They are either methylated, unmodified, or acetylated, and they can transition in between these 3 states. Now, you can see that there are 2 types of transitions here. There are just the usual ones, the straight lines, and then there are these sort of strange feedbacks that you're seeing here. So essentially what this means in terms of mathematics, in a fully deterministic model, where we are essentially modeling the concentration of acetylated histones over time and the concentration of methylated histones over time, you have linear terms that correspond to the straight lines, and then you've got quadratic terms that correspond to these feedbacks. And essentially these feedbacks are quadratic because they are thought to be modeling the effect of these reader-writer enzymes. So for example, if we take the example of this feedback from an acetylated to an unmodified nucleosome due to a methylated one, essentially means that an enzyme is binding to the methylated nucleosome. That's why the feedback has some CM there, but also then it's deacetylating this acetylated nucleosome. So essentially that's why it's quadratic, and it's composed of the product of the substrate times the product of the probability, the enzyme being there. And well, this is a fully deterministic model. For simplicity, we took it symmetric, and essentially you can see that it's bistable due to these feedbacks. And this is the takeaway from this slide. Due to these read-write feedbacks, we can get an ODE system that is bistable. And we actually, because this is a very symmetric and simplified case, we can actually compute easily which are the stable steady states and actually also the unstable one, and even the separatrix, which are the 2 basins of attraction of these fixed points. But all of this was fully deterministic. And if we want to model switching, then we need to, and especially switching due to replication, we need to model replication and the stochasticity that comes with it. So essentially, in our phase space, it would be, we want to model this sort of the effect of replication. How does replication from a fixed point will essentially take you to a different point in the phase space. And the usual way to model this, the vanilla flavored hypothesis, is just to assume that if you have n nucleosomes, each nucleosome is going to be inherited by a given daughter cell with a probability of 50%. So essentially, you can model that with a binomial distribution. Now, because we were working with ODEs and continuous distribution, we took a normal approximation of this binomial distribution. And essentially, now you can compute the switching rates. And the switching rates is just essentially the integral of this probability distribution function over the basin of attraction of the other fixed point. So essentially, how likely are we of going from this fixed point to somewhere in this green triangle here? Now, integrating this, we can actually obtain a fairly nice approximation, which is in terms of this error function that has two terms in its argument. It has the n, the number of nucleosomes, but also this C- and C+ that essentially represents where the fixed point in our model was. And well, this is a very simple model and all models are wrong, but some of them are useful. So the question here is, can this simple model actually be useful? Can it help us understanding some of the data? So, well, thankfully, as I was saying, there is some data for this particular locus in fishing yeast. And they did a very nice set of experiments when they actually measured the switching rates from the active to the inactive state in the, say, wild-type locus, but also in other loci where they actually inserted DNA into the locus. So essentially, they made the locus larger. Now, essentially, our model also was giving us some sort of prediction of what would happen here, because this error function, as I said, had 2 parts. One of them related to nucleosomes, essentially n. Here, in the case of the experiment, it would relate to length. The longer the locus, the more nucleosomes there will be. So essentially, our model was making a prediction of how this should behave. And then C- and C+ would depend on the fixed points, or essentially dynamics of the system. Now, that is hard to estimate because essentially it amounts to estimating what are the rates of the biochemical reactions here. So essentially, we just took it as a free parameter, and then we compared with the experimental data. And here you can see experimental data in orange and the model in blue. And you can see that actually the agreement is not good at all. I would consider this a failure. Now, the question is why? Why was this a failure? Well, one of the reasons could be that essentially we were just considering that the acetylation, methylation, these fixed points, couldn't be modeled by a single free parameter. While maybe that is not true, and that might not be true because when you are inserting DNA, you are essentially changing somehow the characteristics of the locus. Because as I was saying before, SENH is a region that actually helps the silencing the locus. But when you insert DNA, you are inserting neutral DNA that is not more prone to silencing or expression of the gene. So actually what we thought, okay, maybe we cannot model it just with a parameter. We actually need somehow something that amounts for the dilution of these particular sequences of the locus that essentially drives the silencing. And when you enlarge the locus, it makes it less likely to become silent. And now with these two parameters, then we could actually get a much nicer fit to the experimental data. And essentially, this was hinting at the fact that maybe this dilution of the particular sequences in the locus of interest actually is an important effect that should be taken into account. And yeah, I just want to finish with this slide. There's a lot going on, but I won't have time to explain it. But I just wanted to say that in our collaboration with Daniel Saxton and Jasper Rijn, we actually did a very similar study, but in this case in budding yeast. And we actually find a similar trend for the dilution of these genetic sequences when you make the locus larger. And here, in this case, the genetic sequences that were diluted were the silencers. And importantly also, this was a fully stochastic model, and still we were able to obtain to see two different types of transitions. One that was noise-driven, that was essentially due to the noise in the biochemical reactions, and another one that was clearly replication-driven. Yeah, and essentially you can see this in these histograms because you can see that in the replication-driven one, the effect of replication was crucial for the silencing to occur. And with that, I would just like to finish and yeah, leave you with the two takeaways of the talk. That is that there is noise at replication, but it becomes reduced for larger loci, as you can see in the function here. But also, when you are looking at the size of these genomic regions, you should take into account that maybe when making them larger, you are diluting away the effect of specific sequences. And I would just like to thank the Howard Lab, especially Martin, our collaborators Jasper Rijn and Daniel Saxton, and you for your attention.
Thanks, Ander, for the very nice and clear presentation. So is there any question?
I can. You want to go ahead first, Maria? Or okay. I had a question, Ander. There's a really nice talk about the last slide you showed when you had all those different graphs. So I noticed there was
sorry, that wasn't very clear.
This looked very cool, and I was just wondering if you could go more into detail about it, and specifically about these error bars on some of the experimental data seem really large. So yeah, how should we interpret that?
So the thing is that originally there were two. Well, I mean, you should interpret it in the sense that these are unlikely events. So for example, here you can see that it's 2%. So essentially you have to let it grow for 50 generations to actually see one of these events. So they have and they are quite noisy. And the thing that here we are also mixing two different experimental data sets. One that they were done in the same lab, but there still were, for some reason, quite noisy. We don't really, I mean, different people doing the experiments, biology is messy. And yeah, but essentially here we wanted to prove, we actually managed to convince them to obtain this new experimental data because we wanted to prove that there was actually a trend here in the silencing establishment rate that would actually sort of prove the fact that the effect of these silencers, when you're making the locus larger, yes, they are being diluted away. So essentially, you're becoming less able to establish silencing. And essentially, in their original data set, there was a bit of a trend in the red data, but they claimed, and it was not statistically significant. And it was only when we actually took it to much larger data, much larger loci, well, slightly larger loci, that we actually got a significant drop that essentially would prove the fact that, yeah, there is something that is essentially, and we think this is the insertion of DNA that is making these genetic features essentially become diluted and less efficient in establishing silencing.
Yeah, the two samples at the 12 nucleosome number line up pretty well. It seems like, at least for that one, there's not a lot of variation between the 2 years of data.
Yeah, but in the other one, the silencing loss, for some reason, I cannot give you a better answer. Neither could Daniel or Jasper. Yeah.
Well, thank you.
It's what it is.
Okay, we have still some time for a very quick question, if someone wants to ask one. Otherwise, we can thank the speaker again. Thank you, Ander, for the very lovely presentation. And we can move on to our next speaker. So our next speaker is Adam MacLean from the University of Southern California. Is a PI at the Department of Quantitative and Computational Biology. His research spans several scales in biology. So it goes from single cell fate transition to multi-scale tissue dynamics. So some examples of these multi-scale models include kidney branching, organogenesis, and inflammation-induced tumorigenesis. But today, I think it will focus at the smaller scale, so at the single cell level. So Adam, the floor is yours whenever you want to start.
Thank you. Hello, everyone. My name is Adam MacLean. I'd like to thank the organizers for the opportunity to speak. I'm very excited for CDEV Fest. And I'd like to give a special shout out to Maria, who I think has a truly exceptional ability to bring us together in these communities and to make events happen. So I'm really excited to share some recent work from my lab on modeling development from single cells on clonal fates and transition states. Let's get right into it. My lab works on cell fate decision-making. So to bring us all onto the same page, I'd like to begin with a brief survey of cell fates. That is cell types, what a cell can do. So this is my cell, and its first fate is to do nothing. This can be an active choice and a very important one. Its second fate is to divide. This is, of course, fundamental to all of life. My cell can also die. This can also be an active choice and one which is crucial to the establishment of multicellular organisms, and it can migrate. The cell can also transform from one state to another. And finally, it can differentiate through a combination of the processes of division and cell state transformation. And these cell fates, I mean, I think they're really fascinating in and of themselves. I love studying them, but they also underlie hugely kind of important and impactful topics, everything from the development of an organism to its homeostasis of organs throughout life, and kind of underlying also many diseases, including cancer. So if those are cell fates, then what is a cell state? Well, I'm going to pass the buck slightly and say everything else. This is a figure from a perspective article I wrote with Tian a few years ago, where we looked at the existence of these, what we call intermediate cell states from single cell data. And what we're looking for is cell states which are in some sense intermediate. That may be temporally, it may be spatially, and we can identify them through some means of looking at the cell state landscape. So if you're used to kind of, or able to think about a pseudo-potential landscape, then you can think about these cell states as shallow attractors or non-fixed points on this landscape. They're not the deep wells, which we refer to as the cell fates. Okay, so this may be a cell state, a shallow attractor well, or perhaps here, which is not a fixed point. Okay, but not this one. This is my cell fate. Okay. So moving on then, I'd like to talk about the dynamics of EMT. This is the epithelial to mesenchymal transition, by which these epithelial sticky cells that form the lining of your organs, they transform into a mesenchymal state, which is highly mobile and motile, able to move around the body. It happens in development and in wound healing, and cancer co-opts it for its own good. It's a primary cause of metastasis. So we must understand, and we want to be able to control it. And we think that intermediate to these E and M states, there are intermediate cell states which are really crucial, perhaps the most important for metastasis. So this is greatly, well, highly studied system, but our approach is slightly different in that we're really interested in digging into more detail through single cell gene expression data on the dynamics of EMT. So I'm showing you here two trajectories which differ not in their final and start E and M states, but in their transitory states. And so you can imagine different timings, different quantities of these intermediate states. You can also imagine different temporal start and end points. Okay, again, the E and the M states are not different, but their number and their initiation, or the point at which they arise, can change. So a PhD student in my lab, Mei set out on a pan-cancer quantification of these EMT dynamics. What I'm showing you here are a lot of different data sets, all analyzed by single-cell RNA sequencing, different tumor types, different stimuli. And we're plotting the distribution of EMT scores from E states on the left to M states on the right. And to a more or lesser extent, all of these data sets in which we observed an EMT. So there was some sort of spread in the distribution of scores, a transition from E to M. But you can, of course, here appreciate that they have different start and end states and ranges of scores. This led directly to our question here, which is, can we find markers of the dynamics of EMT? I'm going to show you what we mean. So when you think about markers, when we think about kind of traditional data analysis, you could divide up your cells. This could be bulk or single cell sequencing, and then perform some enrichment, some differential gene expression. Okay, so that's a very standard analysis. And we did start there and look at these marker genes for transition states, inferred directly from data. So we had these many data sets where we all clustered through unsupervised means to identify the intermediate states, the orange cells here, and then we look for what genes are upregulated in those. We can also do this via RNA velocity, which is also looking for genes which are upregulated. Now we're looking locally and using velocity vectors at the kind of like genes which are upregulated in a given sample. And we can also find kind of consensus genes across many data sets. But we were really interested in going a bit further and looking for genes that were associated with rates of transition. So to do that, we wrote down a very simple ODE model of EMT, 3 species, the E, the intermediate state I, and the M state, with 2 parameters controlling the transitions, K1 from E to I and K2 from I to M. And now we're going to rearrange our single cell gene expression data, plot these over pseudotime, which defines the axis from E to M. And then we're going to kind of count these cells in bins over pseudotime so that we can get kind of pseudo counts, proportions of cells in each population over this temporal axis. And with this, we're going to fit the model to each individual data set via Bayesian parameter inference. I'm showing you fits from 4 of the kind of 12 data sets we fitted here on the right, and you can see that the correspondence between the data points and the curves, which are samples from the posterior, are very good. So we're able to fit these models, and what we get out is a quantification of the size and the rates of transition into and out of the intermediate state. So we can visualize this all together through the posterior parameter distribution for these 2 parameters, K1 and K2. And you can see global trends, such as an inverse proportional relationship as we transit through the EMT. And you can also see kind of local properties of this plot, like if the cells from different samples were stimulated by the same stimulus, TGF-beta or ZEB1, then they're likely to lie together in different regions of this plot. This gave us encouragement that we're capturing some general properties of the transition rates of EMT that may be interpretable across data sets. So we went on because we really wanted to line up genes that associate with these dynamic rates. So what we did was look here using experiment correlation at the relationship between the posterior parameters, the maximum a posteriori values, with the log fold chains of individual genes across many data sets. I'm showing you a few examples. Of course, many gene parameter pairs were completely uncorrelated, and then some we found evidence that there was a correlation. And again, this is over many data sets, so we're looking for robustness in these phenotypes. And what came out were a few top candidate regulators of these EMD dynamic rates. Oh, here I'm just showing you that the vast majority of these genes are not in canonical EMT databases for genes, of which there are many. So we really are finding new genes, at least not previously associated with EMT, and they're enriched for biological processes associated with the plasma membrane and the cell junction. When we look at these, a couple of our top candidate regulators include NRG1 and SFN. These are two genes which were both kind of significantly found to drive cells into the intermediate state. And I'm also showing you the log fold changes, and in the case of NRG1, the RNA velocity, which was also significantly regulated in the intermediate state. So we're kind of excited about this pipeline in principle, and also these specific predictions. These are both genes that had controversial literature where some people said, oh, it promotes the EMT forwards, and then other studies disagreed and said, no, it supports a reverse transition, EMT transition. And I think that this can really be resolved if it's actually associated with the dynamical process itself rather than a fixed cell state. And we're lucky to have kind of strong collaborators in the Cancer Center here at USC, where we're kind of experimentally testing these genes right now. So I'd like to move on and talk about methods that we've introduced to infer cell fates now over much longer timescales, like not the process of cell state transition, but say a whole lifetime. And this was really started by this observation, which is really stark, which is that for individuals under 65 years old, your blood is massively polyclonal, which means it's just made up from clones derived from many, many stem cells. That changes sharply when you're about 65. And that's true in healthy individuals. This is not related necessarily to disease. And what happens is the blood becomes clonal. Somatic evolution has occurred. Of course, this is commonly studied in cancer, but I'm talking about somatic evolution in your healthy stem cells. And we see this pervasive arise of what's called clonal hematopoiesis, which is just a loss of clonal diversity in your blood stem cell pool. The confusing thing is there's very often no driver mutations to be found. So where is this variation arising to, and how is this somatic evolution occurring if there are no driver mutations? But there's two key technologies that we kind of utilized here to make discovery into the variation and the origins of variation in blood stem cells. The first is this idea of fluctuating methylation, as illustrated by Jay, who's postdoc Jesse's dog, and he's fluctuating back and forth from two states, which is exactly what some CpG sites do on the genome, which we call neutral sites. So these are in no known regions of active regulation, and we can use them as barcodes, lineage markers for stem cells. And the great thing is that we can see these very early, even in utero and before birth, where there's not enough DNA mutations to be able to do cellular barcoding that way. And secondly, we have this kind of blood and stem cell data from twin studies. And this gives us a unique opportunity to also look at variation that arises before birth, because monozygotic and dizygotic twins both share genetic components, and monozygotic twins can also share blood circulation in utero. So we did all this. We kind of analyzed the fluctuating methylation data at these neutral sites. I'm showing you all the data here from birth until old age for the three set comparisons, the monozygotic, the dizygotic twins, and the unrelated individuals. And you can see that kind of there's obviously a lot of overlap, but these curves are different. And strikingly, some monozygotic twins are highly correlated blood composition late in life. And you can see they're also different at birth. So this gives us a hint that already we need some variation acquired before birth. To study that further and to really answer the question, how does blood stem cell variation arise, we developed a single cell model of fluctuating methylation dynamics in stem cells. And essentially, this model consists just of two processes: cell replacement, which is a Moran process, and then we have the fluctuating methylation, the flip-flopping between individual sites, which I'm showing you here at the top. So moving on, then, we can simulate development. And starting from a small number of cells with equal number of clones, we grow this up to the full size of the stem cell pool with frequency-dependent growth, which leads to variation in the clonal distributions, which we saw was able to match the data on twins and unrelated individuals. I'll just show you the start of that. It's on a semi-log scale, so it slows down. We start from equal numbers of clones, and then they start to change in distribution and frequency as the stem cell pool grows. Now we're going to move on from birth and model the turnover of the blood stem cell compartment throughout life with some selection. So selection, just coefficients are sampled from a gamma distribution with either strong or weak parameters. Here I'm showing you an example of weak selection in a monozygotic twin pair. So I'm showing you the clonal distributions on the left, the Pearson correlation between the methylation distributions, and then on the right we have the individual two methylation distributions for the two individuals. So we can see a hint that this seems consistent with the data. Of course, not always. Sometimes a single clone takes over. The nice thing about this very simple methylation readout is that we can see this, right? Even though it's a very simple readout, we don't have resolution at the single cells or even the single clones. If a clone takes over, for example, we get this completely divergent methylation distribution in the data, which we can see, a loss of clonal diversity. So now, when we looked at the many kind of simulations over lifetime under these three models, neutral, weak, or strong selection, we found that neutral dynamics were not really consistent, were a good fit for the twins pairs, that they were too uncorrelated throughout life. And we see that strong selection is, well, actually not a good fit for any model, but especially true for unrelated individuals, where we're still seeing some of these kind of same clones take over, which we know should not happen from what we see in the data. So looking then at weak selection, this seems to be the most consistent. And we see when we look at all three of the different twin samples with weak selection, that the results are consistent with the data, kind of leading us to this final slide, which is just summarizing. These are averages over 100 simulations per condition. So we haven't changed any parameters except the twins' development status. And we can find this consistent model driven by weak selection with variants arising during development. And they're able to describe the lifetime stem cell dynamics with no driver mutations at all. Okay, I'd like to summarize. States, fates, and their dynamics can be inferred from these high throughput sequencing data. I talked about pan-cancer analysis of EMT and how that enabled the rates of inference of the rates of cell state transitions. And through FIPS, the model, we revealed genes associated with the dynamics of EMT. We also looked at a model of clonal dynamics of blood stem cells and how it captured the development of twins versus unrelated individuals. And using this model, we're able to show that variants arising during development under weak selection can explain clonal hematopoiesis that emerges much later in life. Just before I end, I'd like to give a call out to the CDEV Fest community that we really need models more than ever in the kind of my field, which is looking at big single cell genomics data sets where we have just tons of cells annotated into cell types, perhaps, but hugely high dimensional and rich and messy. And it goes from yikes to like, oh dear, whoa, to ah. And, you know, it's like, you know, this is such a rich, yes, scary, yes, bioinformatics is essential, but this is also such a rich place to come in and look for the right means to reduce this dimension way, way, way down to something lovely and nice that we can model. So, you know, if you're looking for some systems, then I highly recommend it. With that, I'll stop and thank everyone who contributed to this work. The EMT project is the work of PhD student Maylee McDermott in my lab, and the blood stem cell dynamics is the work of postdoc Jesse Kreger in my lab. I'd also give a shout out to their important research assistants for Jesse and for Maylee. Thank you very much.
Thanks a lot, Adam, for the very nice and entertaining presentation. So, is there any question from the audience?
Can I ask a hopefully quick one?
Yes.
Adam, I was really struck by that little model of the EMT transition because every term is second order. So, and unless I misinterpreted the terms, it looks like each state recruits cells from the prior state. So in other words, you know, the I cells recruit E cells and so on. What's the biology behind that?
Yeah, the EMT model we write down is like as a exactly kind of, you know, almost a kind of very simple enzyme dynamics type of affair. I mean, essentially, we wrote down the simplest thing we could so that we could see these basically compare these relative rates. Yeah. So the second order terms basically help with what we see in the data, which are, you know, transitions through. And it's important to remember, like when I'm talking about rates, that like everything has very weird units here because I'm fitting cell proportions over like pseudo time. So like I certainly, like, you cannot, these are not rates which have biophysical units that I can go away and measure, but we're taking these rates, like we're always going to like kind of look at the relative comparison to one rate over another and ask how they change relative to one another.
Okay.
Alessandra, you're muted.
Sorry.
And there's a couple questions in the chat.
Yes.
That's what you're saying.
Sure.
Yeah. So I'm going to read a couple of quick questions from the chat. So Leah is asking, does age 65 emerge from some parameters, or what accounts for that age of transition to monoclonal?
Yeah, so the age 65, this is not a model. This is all data. So it's, you know, and it's not like I'm not talking about some very sharp transition, but you do see the kind of like around about then, like a tipping point with quite a very clear threshold in the kind of those pictures I showed you of the kind of the clonal phylogenies of the bone marrow, where you just see, not in all individuals, but in, let's say, like many healthy individuals become have these subclonal growths, which, you know, are associated with cancer risk, with risk of diseases in the bone marrow, and some individuals do have what we call a kind of CHIP, clonal hematopoiesis of indeterminate potential, which can develop into blood diseases. But we also see it in many healthy individuals. But no, there's no model required for that. It's just what we see in the data.
Okay. I'll just read the final quick question. Victoria is asking, for transition states, how can one distinguish between the existence of the shallow attractor and the slow manifold no attractor?
Yeah, that's a really nice question. So I guess I can think about like in this question in several different ways, like from a perspective of biology or mathematics. I mean, of course, you know, mathematically it's obvious, but I think biologically in many types of the systems where I'm asking questions about, it might be like kind of functionally not possible to distinguish between those two. And it is a good point. I would say that I tend to be very pragmatic about these things, and I probably don't worry that I can't tell those apart. And the reason why is that they're, you know, it's in any case we never have, I mean, what I presented to you in the EMT project, at least, was a deterministic system. I mean, we always have noise, and we have, so in reality, we have the ability to switch even between attractors, which you can't necessarily switch between in a deterministic system. So yes, the answer is that, like in the case of the biology, I think it might be practically very hard to tell these apart. But it could be interesting in some cases to look for kind of like sufficient conditions to discern.
Thanks, Adam. Thanks for the presentation and answering all the questions.
Thank you.
So we welcome our next speaker. That is, I apologize if I mispronounce the name, is Shibashis, who is a postdoctoral researcher at the Laboratory of Computational Systems Biology at the University of Tennessee, is interested in exploring a variety of dynamical phenomena in the context of system biology using computational and analytical tools from nonlinear dynamics. So we will hear a story about gene silencing and the modeling of such process. So the floor is yours whenever you're ready. I think you're sharing the presentation mode, not your slides.
Okay. Is it?
Now we
it-
just see the list of your. Okay, perfect.
Yeah.
Thanks a lot.
So, hello everyone. I am Shibashis, and I am a postdoc at UT Knoxville. And today I will talk about a continuum model that we have developed to investigate the process of X chromosome inactivation. So, first and foremost, what is X chromosome inactivation? So, X chromosome inactivation is a fascinating biological phenomena that equalizes X chromosome X-linked gene expression in male and female cells. And it works like a molecular switch by deactivating the redundant X chromosomes present in female cells. And having a clear picture of this process is actually very very important to understand the whole process of gene silencing. So during X chromosome inactivation, one of the two chromosomes present in each female cells gets deactivated in a random manner, and this randomness is manifested beautifully in tricolored calico cats, where depending upon which gene, which X chromosome the cell decides to keep active, the far takes either black or orange coloration. So now we have to discuss the biochemical mechanism of X chromosome inactivation, which is orchestrated by a long non-coding RNA called X inactive specific transcript, and in short, XIST. So what happens is during the early phase of X chromosome inactivation, the XIST RNA molecules gets transcripted from the X inactivation center of the chromosome and spreads throughout the chromosome and gets tethered to certain regions of the chromosome architecture. And after tethering, the XIST molecules recruits a protein complex called polycomb repressive complex and silence the genes by methylation of nucleosomes. So this is the overall biochemical picture of X chromosome inactivation. And this is a schematic representation of the same to get things much more clearly. So what happens at the early phase of X chromosome inactivation, the XIST molecules, which are represented by these green lines, are getting transcribed from the X inactivation center, and they spread throughout the chromosome architecture, and some of them get tethered to the X chromosome, which is represented by these blue lines. And finally, we recruit the polycomb repressive complex and carry out the gene silencing, which is represented by these red shadow regions. So this is the overall rough picture of X chromosome inactivation. Now, conventionally, the process of X chromosome inactivation has been modeled by a particle-based statistical framework. For example, this is a very recent study published in 2021, where they utilize this type of particle-based statistical model to study this X chromosome inactivation using very simple kinetics and statistical reaction diffusion simulations. And these type of study are very revealing in terms of especially experimentally inaccessible length and time scales. But however, there remains certain biochemical features of this X chromosome inactivation, which are very difficult to capture by these frameworks. Now, one such is the biochemical feedback mechanisms involved in the process. So what are the specific biochemical feedbacks involved in the process of X chromosome inactivation? So recent experimental studies actually reveal that the production and degradation of exist molecules itself are autoregulated, and moreover, the spatiotemporal dynamics of active PRC2, which is responsible for chromosomal inactivation, is dependent on the positive feedback that is imparted by the neighboring epigenetic markers of the inactivation site. So these are the feedback mechanisms, important feedback mechanisms involved in the X chromosome inactivation. So to include those feedback mechanisms into our modeling scheme, we have utilized the partial differential equation based reaction diffusion framework. And as usual, we had the few advantages, which is it is easy to implement complex reactions and regulatory networks, and it is very computationally inexpensive. But apart from that, we had the usual disadvantages as well. The striking one was the difficulty to describe complex geometries. Nevertheless, for our 3D PD model, we have devised a way to include semi-empirical chromosomal structure derived from high-C data. Now, we actually addressed our modeling in a segmental way. First of all, we actually addressed the exist production and degradation, and then we addressed the X chromosome inactivation module. And finally, we join these two modules through a third module called exist deadening module. So we will come back to it later and have a much more clear view. It is a bit puzzling right now. So these are the partial differential equations that describe the production of exist and degradation of exist, and this is the spatial diffusion of exist molecules. And this is the partial differential equations that describe the X chromosome inactivation, where U1 is proportional to the chromosomal inactivation because it represents the active PRC2 complex. So this is the whole picture, and as we can see here that these Hill equations actually representing the biochemical feedback mechanisms that were included in the process of X chromosome inactivation. So there are three key parameters that we have studied and they are the production feedback coefficient of exist, the degradation feedback coefficient of exist, and the positive feedback coefficient imparted by the inactive X chromosome. That is the neighboring epigenetic markers on the process of X chromosome inactivation. So now, coming to the exist spreading module, we first ask the question whether there is an optimal feedback strength for fastest maximum spreading of exist molecules. To do that, we actually simulated the spatiotemporal dynamics of exist molecules for each of the parameter sets, and from the simulation result, we derived a bubble, and the radius of the bubble is actually proportional to the extent of spread, as you can see, with more homogeneous spreading. the bubble is getting bigger, and the brightness of the color of the bubble is dependent on the speed of the spread. So what we are looking for in other terms is that a sufficiently large bubble with bright color that actually represents the fastest maximum spreading of X-cyst. And the scan actually reveals that indeed there is an optimal feedback strength for which the fastest maximum spreading is observed. And this actually also reveals that the feedback mechanisms actually play a pivotal role in the spatiotemporal dynamics of X chromosome, X-cyst molecules. So now we come to the X chromosome inactivation module and wave pinning. So why wave pinning is important? It has been long established that although the X chromosome, inactive X chromosome remains inactive, there are almost 20% of genes which are escaped from the process of inactivation and remains active. The long non-coding RNA X-cyst is a very striking example of that, that it is getting transcripted from those SKP genes. So to account for that SKP genes, we have to include this wave pinning feature into our model. Otherwise, the whole chromosome will get deactivated, which is not desirable. So we ask the question whether it is at all possible to achieve this wave pinning into our PD modeling framework. To do that, we actually sample a tiny region from X chromosome in 1D. And for an arbitrary parameter set, we could see that this homogeneous inactivation is taking place. And by modulating the spatial parameters, we could achieve this type of artificial wave pinning, which might be prudent biochemically, but not so interesting from a modeler's perspective. So after a lot of search and motivated from this study published in 2008 from Professor Keshet's research group, we actually able to achieve this type of wave pinning phenomenon into our model. This actually reveals that intrinsically it is possible to achieve this heterogeneous inactivation in the model. So now we finally ask the question, what is the effect of feedback coefficient on the process of wave pinning? To do that, we actually use the same approach in 1D and validate the feedback coefficient from a low value to a high value. And the simulation actually reveals that indeed the feedback with increasing feedback coefficient, we are able to get this varying extent of X chromosome inactivation, which is also observed in different tissues as well as when the transgene X is placed on the autosomes. Now, finally, we bridge the two modules through this X-cyst tethering module. So why this X-cyst tethering module is important? Because X-cyst is present in the system in a very low number. So one might ask the question why you are modeling this in a continuous manner, because X-cyst is so small in the system. So to include that stochasticity in the picture, we actually use this X-cyst tethering module so that this continuous nature of X-cyst is converted to a stochastic nature by this, like this phenomenon. So this is the overall picture in the combined module, where finally we get this type of heterogeneous inactivation of X chromosome. Now, finally, we actually use this experimentally, use this IC experimental data and derived this average chromosomal structure, and this is a semi-empirical average chromosomal structure which was reported in this paper published in 2021, and this structure is actually revealing the chromosomal architecture, which is keep in mind is a average structure, not the actual structure. This is achieved by cross-linking the simulating a Gaussian chain and cross-linking the result with the actual experimental IC data. So now, so these are the tethering steps, and this animation actually revealing the spatiotemporal dynamics of the X chromosome inactivation. And we can see that in the steady state, the heterogeneous X chromosomal inactivation is achieved, and more importantly, the escaped genes are residing in the peripheral region of the chromosomal architecture, which is also in line with the experimental findings. So in summary, I want to point out the PD model indeed captures several key biochemical features of the chromosome inactivation. And secondly, the autoregulation has turned out to be a determining factor for spatiotemporal profile of X molecules. The extent of X chromosome inactivation is determined by the feedback imposed by the inactive X chromosome, and 3D simulation reveals the peripheral positioning of the escaped genes in inactive X chromosome. So I want to acknowledge my advisor, Jan Hong, and my collaborators for useful discussion, and the funding agencies. Thank you. I would like to have some questions.
Thanks a lot for the interesting insights in gene science. So is there any question from the audience? So I can see a raised hand by Lie. So if you want to unmute yourself and ask the question.
Yeah, hi. Thank you for the very interesting talk. So I don't know exactly what PRC is, but is there some reason why the active PRC would diffuse more slowly than the inactive? Than the inactive PRC?
Because exist is active PRC means that those active PRCs are tethered to actually these active PRCs are tethered to these exist molecules. So now, as the exist molecules are tethered to the chromosome, so they are moving much more slower than the free PRC2 complexes. So that is why we are also implemented in our model as well. The diffusion coefficient of active PRC2 should be much more lower than the inactive PRC2.
Thank you.
Thank you. Is there any other question?
Can I ask one?
Yes, please.
Sure. I was wondering whether, thank you very much for that talk. I was wondering whether your model actually says something about why one of the X chromosomes is inactivated or not, whether you're actually implementing some sort of PRC sequestration process or, I'm not sure if you explained that, but I was just wondering because many other models have some sort of coarsening dynamics that help explaining that feature.
Yeah, I know, but for our model, we are actually starting from the point where one of the X chromosome are getting chosen. So yeah, you are right that some of the model actually include that type of selection process, but no, we haven't done it. We are much more interested in the effect of biochemical feedback on the process of on this chromosomal inactivation process. So yeah, so that's
thank you.
Yeah.
So let's thank our speaker again. And now I'll leave the floor to Maria to introduce our next speaker, Ruth Baker.
Hi, everyone. Thank you so much for the great talks so far. Well, I have the pleasure to introduce Ruth Baker today. She's a professor at Oxford, but her team uses math, computation, stats, and other methods just to study cell and developmental biology. She is currently interested in cell motility and how cells move collectively in the developing embryo. But really, Ruth has like an outstanding contribution to the mathematical biology field as a whole. And I honestly urge everyone who is currently curious where the field is heading to look at her work. So today she will discuss her work on mechanical constraints in cell cycle regulation in models of collective cell migration. Ruth, the floor is yours.
Thank you. Can you hear me okay? Yeah, well, and thanks for the introduction. I'm not sure I can live up to that, but I'll do my best. And this talk comes from me and my cat. So Adam's was present for the maths, I think, and mine likes to be front and center stage for the actual talk. So what I want to talk about today, I want to talk about some work that we have done, which is kind of partly theory and partly, I think, some biological insight that relates to the extent to which cell motility and the cell cycle regulation through mechanical constraints sort of impact and play a role in cell invasion. I haven't really given all of this talk before, so bear with me and please ask questions if anything isn't clear. So the picture you were seeing on the front slide, published by Saito and Imamura in 2015, is a picture of an early stage zebrafish embryo developing. And in this system, what's been done is that the authors have used something called the Fuji reporter to label the cell cycle. So for those of you who aren't familiar with this, the kind of basic story is that if cells are in G1 phase of the cell cycle, then they express a red fluorescent protein. And then in SG2M, they express a green fluorescent protein, and then just after mitosis, they go dark for a short period of time. And so we're interested in understanding, I guess, the extent to which having information from this Fuji reporter can help us probe sort of cell cycle regulation. For example, how motility might depend on the stage of the cell cycle that a cell is in, and how that all combines to regulate collective invasion. And I think, yeah, I'll say a bit more about how we do that, I guess, as we go. But I guess the sort of very simple idea behind building models of this system is that you take a cell that's initially red in G1, it eventually transitions to SG2M, and then when it divides, it produces two new daughter cells that eventually then enter G1, and the process repeats again. Okay, so I think this reporter is really, really useful, and one of the things that we wanted to do is to think about how to build a mathematical framework to learn about collective invasion using some in vitro assays where essentially they're quite easy to conduct. They're still used really widely and at scale for things like sort of drug testing and looking at gene knockdown studies. But for our intents and purposes, they're simple enough, and we can collect lots of data so we can visualize where every cell is in the assay. So the two main data sets I'm going to talk about today are one which comes from a barrier assay. And so in this assay, you essentially initialize cells, to describe it kind of within a cookie cutter, you grow them to confluence, and then at sort of time 0, you remove that barrier, that cookie cutter, and you essentially watch as the cell population grows and expands, migrates into the available space. So that's a barrier assay, and the other one is a scratch assay, where you essentially grow cells to confluence in a dish, and then at time t0 you take a pipette, you essentially remove a portion of the cells in the center of the dish, and then you watch as the cells close and fill that available space. And so both of these, the data that we've got here, we can see very clearly the cells that are in sort of G1 and SG2M phase. So what are the questions? So I think I've said it already. What can we learn about how the cell cycle is regulated during collective invasion, essentially by mechanical feedback, and how the cell motility might be regulated? And can we do that through combining quite simple models with these data sets? But I said it was a bit of theory, and so there's some questions that I want to ask along the way. So I think one of the big questions for mathematical modelers is to ask, how do I know when my model is at quote unquote the right level for the data? And I'm going to try and access through that question by thinking about, are the parameters of my model identifiable given the data? And if they are, can I estimate model parameters and quantify the uncertainty of having those estimates? And I think that for me, the really important part about, and quantifying uncertainty is then understanding how that uncertainty in parameter estimates propagates forward into the uncertainty in model prediction. So what are the range of expected outcomes I might predict? So for this talk, it's going to be quite simple. It's going to be models that are systems of PDEs, in fact, at most 2 PDEs. And I'm going to be often interested in parameter inference using a Bayesian framework to quantify uncertainty. So I want to access the probability, the posterior probability of model parameters theta given data D, and access that in terms of the product of the likelihood of the data given the parameters and the prior distribution. Okay, so where are we going to start? We're going to start by thinking about what structural identifiability is. So this is something that's a property essentially of the model, nothing to do with the data at this point. And it really just asks the question, if you write down a model, will distinct parameter sets for your model always give rise to distinct distributions of observations? And if that is indeed the case, then we call our model structurally identifiable. And that means that in theory, it's possible to determine the values of the model parameters from perfect input-output data. So this is something, when I said it was kind of property of the model of the data, it clearly doesn't take into account the limitations imposed by the quality of the data we have. But I think it's still a really important first question. So the first thing I want to spend kind of maybe 5 or 10 minutes talking about today is can we do that for the kinds of models of the experiments I'm talking about? And just to say up front that there's been a lot of work on developing methods for ODE models in this context, but far fewer for PDE models. And I think probably heard a little bit about this yesterday already. So I'm just going to probably not do anything more than that. Okay, so I'm really interested in a simple model that replicates my experimental dynamics. So I'm going to divide my cell population into 2 phenotypes, ones that are basically in G1, so they're fluorescing, expressing that red fluorescent protein, and ones in SG2M, which are appearing green in my assay. And so effectively, both of my populations, I'm going to assume, are just going to diffuse. And they can have non-equal diffusion coefficients, representing the fact that we might be witnessing something akin to the go or grow hypothesis, where essentially cells tend to be far less motile when they're rounding up to divide. So D1 and D2 can be different. And then we have 2 other parameters, very simply, that represent the rates of cycling or moving through the cell cycle. So K1 regulates essentially the amount of time spent in the red phase, if you like, and K2 the amount of time in the green phase. And here we're saying that there's no mechanical feedback on the rate of cycling, which means that these terms are just linear in R and G. And in particular, this factor 2 in the first equation just comes from the fact that when cells divide, they give rise to 2 daughter cells. So we could ask the question, what happens if we assume we measure only the total cell population? So we're only going to measure the amount, the total numbers of red and green cells in the assay. And I want to answer the very simple question, in theory, if that's all we could measure, could we pin down the 4 parameters that we have in this model? And so if you're sort of familiar with how identifiability and structural identifiability works for ODEs, we're going to take exactly the same approach here and aim to reduce the model to what I would call a system of input-output equations in the observed variables. So here I've only got one observed variable and it's the total number of cells. So I want to reduce my system to a single PDE that just in terms of n and its derivatives. And if I can do that, then essentially the coefficients in that equation determine the identifiable parameter combinations. And if you play around with this long enough and maybe use Mathematica because it's a bit easier, what you figure out that you can actually do this in this context. So if Nij is what happens if you take the ith derivative with respect to x and the jth derivative with respect to t, so it's the I plus jth derivative in total, you can essentially write a fairly neat expression that basically just tells you how the total cell density evolves in space and time. Okay. And so this is my input-output equation. Go on, someone have a question? No? Okay, do shout if you do. So once we've got this input-output equation, then what we need to do is divide through by a coefficient to ensure uniqueness. Otherwise, I could multiply the whole thing by any constant parameter and still get the same, infer the same identifiability set of identifiable parameters. So here, trivially, the N02 term is just 1, so I trivially divide by 1 if you like, and then I can see that the set of identifiable coefficients for my model is what's given in brackets on the slide. And so, for example, in here you can see that you can identify, so if we have perfect input-output data, it means that we can have perfect estimates of all of these, or expression estimates for all of these derivatives of n with respect to space and time. And so effectively, if we can get perfect measurements of those derivatives from the data, that means effectively we can estimate these coefficients in this equation. And so in particular, I can get then a handle on the sum of the diffusion coefficients and the product of the diffusion coefficients, which means that effectively I can identify both of those concretely. And in the same way, I can also identify, so one of the coefficients is the product of these rate parameters, and the other one is the sum of one of the others is the sum of the rate parameters, so I can identify both the rate parameters individually. So I'd say that this model is structurally identifiable. So in theory, if I had sufficient data just for the total cell density, I should be able to pin down all of the parameters of my model. So I think that's quite a powerful result, and I think that really I think about this as is structural identifiability kind of being a prerequisite for any model that I want to write down. Okay, so assuming I can observe either one or both populations or the sum of the two, I really want to know whether I've ever got a chance in pinning down my model parameters, and structural identifiability tells me that. It doesn't tell me anything about how much data I need to do this in practice, but I think it's the first step that you need to take. So in this paper, which is will appear in Proceedings of Royal Society A, and you can find a copy on arXiv, what we managed to show is that more generally for coupled systems of reaction, diffusion, advection equations that are linear in the observed variables, then it's always possible to reduce the system to the system of input-output equations and to determine identifiability or structural identifiability, or in fact, what combinations of parameters in the model are structurally identifiable. And I think theory for these more general, what we call semi-linear systems, as well as some mathematical code to help me do this in practice, is provided in the paper. But I think one of the actually just really nice take-home results is that if I consider a couple system of reaction, diffusion, advection equations, so a spatial model and its non-spatial equivalent, where you just get rid of all the spatial terms, then the identifiability, the structural identifiability of that spatial model will always be at least as good, if you like, as its non-spatial equivalent. So essentially introducing spatial effects always, and spatial effects always tells you more. Okay, so in short, that model is structurally identifiable. Okay, so moving on from that, I sort of said, okay, well, what happens when you have actual experimental data. So then you can ask the question, is it possible to provide reasonably precise, where we can define that in whatever way we like to some extent, estimates of our parameters using finite non-ideal data. So this is really like, if you've actually got an experimental data set, how will you do? So one of the approaches we've been taking is to look at what's called the profile likelihood. And so the idea here is that you can write down the normalized, likelihood, sorry, there's a bit of a typo here, the normalized likelihood, which is basically the likelihood, don't read the equations, the likelihood of the data d given the parameters theta, and divide it through by the maximum of that likelihood. So that's a normalized likelihood. And so if you want to think about the profile likelihood, what you do is you take your parameter set theta and partition it into an interest parameter, so typically 1 or maybe 2 parameters if you want a bivariate likelihood, profile likelihood, and call the rest nuisance parameters. And then the profile likelihood for the interest parameter, let's just call it a single parameter phi, is essentially what you get by optimizing the normalized likelihood over all of those nuisance parameter values. And so in this context, non-identifiability looks like a flat likelihood profile. So the likelihood isn't changing much as you scan across different values of this interest parameter and half open confidence intervals, for example, in a range over which you're interested. So let's see what we can do in terms of practically identifying parameters in one of these PDE models. So I'm going to start off by talking about the experimental data from the Scratch assay. So published, I think first published by Vitadello in Biophysical Journal from basically from Matt Simpson's group. And so I think what's quite nice about this data is that if you just look at taking a snapshot across the center of the wound, everything's pseudo one-dimensional, so you can just average the data in the y direction to get essentially a one-dimensional density profile in just X that evolves over time. And this is what that data looks like. So with this data, we were kind of initially interested in asking the question, are the data sufficient to determine whether the cells move with a different speed, so have a different diffusion coefficient in G1 versus SG2M phase? So we'll get to mechanical constraints later. This is just that two cells move differently in different phases of the cell cycle. So we built 2 different models. So the first model, model 1, assumes that cells have different diffusion coefficients in different phases of the cell cycle. So this is really a model that's akin to the previous model I showed you, where we have a D1 and a D2 parameter. And I've just augmented the model a little bit. So this is just sort of the natural model we picked at the time, if you like, to describe these data, by assuming that there's some feedback in terms of the rate of cell division depending on the local cell density. So the model 1 assumes that cells have different diffusion coefficients, and model 2 assumes that cells have the same diffusion coefficient. And so we're really interested in asking to what extent, given the data I just showed you, are these models practically identifiable? So can I confidently estimate the parameters of these different models? And how does my confidence differ maybe between model 1 and model 2? And I think this is kind of a very important and maybe, for want of a better expression, practical question, because we really want to know, given the data we have, what's the kind of limitations that it's going to impose on our ability to predict the tissue dynamics. So in both these cases, to make progress, I'm going to assume that I've got observation error that's normally distributed with 0 mean and some known variance parameter, which is a fairly standard thing to do. And there are obviously other choices you could make, but for now we're going to roll with that. Okay, so let's think about what these profile likelihoods look like. Okay, so I'm going to try and get you to focus for a minute on the upper two profile likelihood. So if I haven't done a very good job of labeling, relabeling, in fact, the axes here, because I realized I changed notation partway through this talk. What I'm trying to show you, so let's focus on this. So this is the profile likelihood for the parameter K1, so ignore the KR, call it K1, that essentially controls the rate of, or the amount of time spent in G1 phase, or the rate of transition from G1 to SG2M. So the profile likelihood says, as we scan over various values of that parameter K1, what's the likelihood? How does the likelihood surface change? Essentially, if we're optimizing all over all of the parameters. And we can essentially also put down confidence intervals there. So this is a 95% confidence interval for this parameter value. And you can see that for this parameter in that data set, we have a very, I think, we can quite precisely, or hope to quite precisely pin down the value of that parameter. So the profile likelihood is nicely peaked at its maximum value. And if you look at K2, so for model 1, this is the on the far right at the top. You can see again that we have a profile likelihood that's nicely peaked and well-defined sort of confidence interval. So again, I call that parameter value or that parameter practically identifiable. On the other hand, if you look at the profile likelihoods for the two diffusion coefficients, so this is D1 and D2, where red and green have the obvious meanings, you can see that you really can't, the profile likelihoods have far different shape, I should say. So they're kind of much broader. The one for the cells in SG2M really doesn't is so broad that we can't really provide a confidence interval here. And that would identify or suggest to us that those parameter values essentially are non-identifiable given these data. So we're not going to be able to confidently estimate them. And on the other hand, if you look at model 2, right, for the two rate constants, things are very, very similar. But when you turn to estimating the single diffusion coefficient, now assuming that they both move with the same diffusion coefficient in both portions of the cell cycle, you can see that you can get a very confident estimate of that parameter value D. So suggesting to us that maybe we might want to think about preferring model 2 as opposed to model 1, because essentially the model is identifiable, and then we can have confident predictions we can confidently make predictions of outside unobserved behaviors using the model and the parameter estimates we've gained. So the profile likelihood approach, I think I'd like to highlight, is really, really useful because you can generate these likelihood profiles in fractions of the amount of time it takes you to generate or estimate the full posterior distribution. But I think that the information you get out is really pretty much the same. So just going through, this is kind of what happens when you use MCMC to generate the posterior distributions. You can see that you get really, really sort of nice posterior distributions, if you like, for the rate parameters for model 2, but you do much less well at identifying the diffusion coefficients, and you see that in terms of the fact that the traces from the chains really kind of bounce around all over the place. And so we'd say, again, consistent, that this model is not practically identifiable. There we go. Whereas for the second model, which assumes the same diffusion coefficients, we see that the chains are nicely well behaved in for all of the parameter values, and we get really nice estimates of the parameters. And I should say, in all of those cases, one of the things you can obviously then do is sample from that posterior distribution and use that to do a posterior predictive check. So to what extent is my data consistent with my model? That's shown down at the bottom there, and things I think look pretty good. So from this part of the talk, which is thinking about practical identifiability, the take-home message is that although both of these models are structurally identifiable, given observations of both the red and the green populations, right, we can see that kind of at a glance using this input-output kind of arguments. The first model that assumes that cells move at different rates in different portions of the cell cycle isn't practically identifiable, so we can't confidently estimate the model parameters. Whereas for the simpler model that assumes we have essentially equal diffusion coefficients, then that is practically identifiable, and we might want to select that one going forwards. Okay, so take-home message: consider essentially the limitations of the data in building the model. And I think identifiability analysis is pretty useful in thinking about what level of model might be relevant for the data you have. Okay, so I want to use these kind of ideas now in anger to think a bit more about the role of mechanical constraints in the cell cycle. So this was a project that kind of came about by happenstance, really. We were sort of, you know, I sort of worked on the first project looking at practical identifiability, and we were discussing with some collaborators at Princeton who have data that, and this is the data, that essentially uses a feature reporter on a bigger tissue that's a much higher density. And we were talking about it, and I kind of, you know, suggested, why don't we try taking the previous model off the shelf and applying to this data set and seeing about what we can learn about mechanical regulation of the cell cycle. And so this data set and our collaborators, in fact, are wonderful, because one of the things that they can do is they can actually identify every cell in the tissue, and they can see whether it's in G1 or SG2M, or in fact post-mitotic. Now, I'm not going to pretend that we've yet made use of all this data, because in fact what we did was just look at then establishing or estimating a radial density profile out from the tissue sensor. But when you do that, one thing that becomes immediately obvious is that the model we worked with in the previous paper just isn't going to cut it. Because the model in the previous paper assumes that all of the cells would get stuck in G2 in the center of the tissue, and in fact that isn't the case. So what it suggested to us was that a model that kind of only has regulation sort of in terms of transitions between stages of the cell cycle. If there's only regulation in one of the phases, you literally cannot capture the data, the dynamics in the data we're showing here. So we thought, what happens if we just kind of augment this previous model just a little bit and assume that there maybe is a checkpoints at both to kind of go between both phases of the cell cycle. So it's the same model, we're using the same diffusion coefficient based on the previous study. And now we just add these terms in, so I'll say again, this F1 term, oops, I'm really good at highlighting, and this F2 term in here, which essentially represent mechanical regulation of transitions through the cell cycle. And we use the simplest possible form for feedback. So if N is the total cell density, then we just assume that the feedback is essentially so the rate of transitions decreases linearly as the cell density increases, and it becomes 0 at some carrying capacity K1 for F1 and at some other carrying capacity K2 for F2N. So this is just saying essentially that we can have mechanical regulation kind of at two places, two checkpoints in the cell cycle, but the strength of that regulation or that feedback might be different at different points in the cell cycle. And again, we just asked the question, can you calibrate this model to the data? Can you confidently estimate the model parameters? In doing so, what might you learn? Okay, sorry. So here is, in short, what we learned. So this is the posterior distribution for each of our model parameters. So on the left, the diffusion coefficient, in the center, the rate constants, and on the right-hand side, the carrying capacities. So I think the first thing that is, I just want to highlight is that you can get from these data, I think, relatively confident estimates of all the model parameters. So certainly we knew it was structurally identifiable. It looks like it's practically identifiable as well. And the second thing is that the model does a pretty good job at predicting the dynamics of the actual data, and it was actually of the dynamics that are kind of in the data itself. I'm not so worried about what's going on behind the front for various reasons, but I think I just want to highlight that I think we can do really quite a good job at predicting the sort of evolution of the cell density at the front, and also essentially the rate of expansion of this tissue. Oh, hang on, get rid of that annotation. Okay, so I guess the next question is what can you do with all of that? So one of the things that you can, in fact, do with all of that is then be able to predict how the length of the different cell cycle phases depends on the local cell density. And in particular, and I guess you can see it in this previous slide, we can see that essentially estimates for the parameter K1 are smaller than estimates for the parameter K2, which then tells you that the checkpoint that regulates transition from kind of G1 to SG2M is a much stronger, or to some extent a stronger checkpoint than the checkpoint that regulates transitions from essentially SG2M to become post-mitotic. So essentially regulates the actual cell division. And to some extent, I think that makes sense because I guess if cells are going to go to all the trouble of replicating DNA and doubling to become able to divide, then they should only do that if they're confident that there's going to be space for the division to actually take place. But in short, we can quantify and predict length of different cell cycle phases, how that depends on cell density, and how then the overall cell cycle depends on cell density. And you can also, these kind of sort of slightly opaque lines show you kind of how the predictions vary as you sample over the posterior distribution. So how much uncertainty we have in terms of our predictions here. So this was really great, but one of the things that we wanted to think about was whether there's actually any evidence for this biologically. Okay, so we're sort of seeing it in the data which suggests it's out there, but has anyone really gone ahead and tested for this? Well, in 2014, as a paper published by Stryker et al., which I'll come back to in a minute, and in that paper, they showed and demonstrated very clearly that there's a checkpoint between G1 and NS phases of the cell cycle. So that checkpoint was kind of shown to exist a while ago, and so we were kind of confident of that one. But it took quite a lot more hunting, a little bit more angst actually on this one, before we could find actually some very recent evidence published in 2022 that says that there's additionally essentially a mechanical G2 checkpoint that's controlling cell division. So this is the additional essentially additional evidence we need that supports the hypothesis coming from our model, which is rather nice. Okay, so I think what we've learned there is that given these kinds of data, it's possible to quantify various aspects of cell behavior, so cell motility, whether that varies, or to determine whether we can see if that varies in various parts of the cell cycle, but also to understand how mechanical constraints impact cell cycle. I think the final point I want to make really is that experimental design is really, really important in all of this. So the data that I talked about by Stryker et al. Just a moment ago that looked at kind of establishing this checkpoint from G1 to S was is pictured here at the top. So they actually used the same, I think, kind of barrier assay, but in that experiment, they grew cells to really, really high density behind the barrier before removing it. And the impact of that is that essentially all of the cells pretty much in that tissue become stuck in G1 phase of the cell cycle. And so when you remove the barrier, what you see is that as cells at the front begin to kind of move into the available space, then that mechanical constraint, if you like, on cycling is lifted. The cells divide, they enter, the cells enter G2 phase of the cell cycle, and that much happened. But because the cell population was at such high density behind the front or within the barrier, you basically see right kind of in the center of the tissue, it takes a very, very long time before you see any cells emerging that begin to cycle again. So had we used this data, I think we'd have got quite different results in terms of seeing these mechanical checkpoints. And the other thing to say is that going back to kind of revisit the data published by Vitadello et al. That we used in the kind of practical identifiability study, we quickly realized that actually that data wasn't really sufficient to pin down the mechanical regulation of the cell cycle because it's initialized at a very low density in this assay. So we've got sort of normalized cell densities that kind of, lying initially below 0.1, which is really quite low. And in fact, if we'd have taken that very first model that I talked about, where there was no mechanical feedback, it was just simply kind of essentially exponential growth in each of the phases, or transitions through each of the phases. And if we fit that model, which is what you can see down here, right, in the colored lines, if we fit that model, it actually does a really great job. So we can identify the parameters of that model and fit it to the data, and it does a great job in replicating the dynamics. So yeah, I think you have to be really careful in the choice of data set when it comes to thinking about what you can actually learn. So from this bit, I've kind of said this a million times, but if we could quantify mechanical regulation of cell cycle, we could see that there are at least 2 checkpoints which are mechanically regulated. And the last point I was making is really that experimental design really matters, and it's important to consider essentially the limitations of the data in building the model. And also that you can often learn new things by exploring the validity of existing models using new data sets. So this whole entire study and the archive references there kind of set about because we were trying to take an off-the-shelf model and interrogate it, or interrogate a new data set using it. Okay, so I don't have much more to say other than thanks for the invitation to speak. This has been a really fun set of talks so far. If you're interested in what we've been doing, I've tried to kind of put the references throughout, but there's a few more here. And I just want to finish up by acknowledging the people who did the work. So here's my group in Oxford, and much of this work was done by Carlos Falco and Yue Liu, who are PhD students in the group, in collaboration with Alex Browning, who's a research fellow in Oxford, Maria Tasker, who was a summer student, and then further afield collaborations with Daniel Cohen and Kevin Su from Princeton, Matt Simpson from QUT, from Oliver McLaren from Auckland. And then just like to finish by saying thanks very much for listening, and I'm happy to take any questions if you have any.
Thank you, Ruth. That was a great talk. We already have a few questions in the chat. Hold on, just let me scroll through.
I can see them, I think. So I think there's one from Adam first, who asked about structural identifiability for nonlinear equations, and definitely. So there's a paper on arXiv by Heather Harrington, Helen Byrne, and some others who have extended this a bit further, and they have, I think, done a much better job of digging into the theory underlying this. So I would recommend just having a look at that. So it definitely works in the nonlinear case. I think where we stopped at was the fact that it becomes non-trivial to think about deriving this input-output equation, or the system of input-output equations. And in fact, in the preprint, we can demonstrate that it's not very hard to break Mathematica and have to wait hours and hours, and maybe 24 hours to generate one of these input-output equations. So I think that's where the barrier to progress is going to be, but I think it can work. I don't really know. I don't have, beyond Heather and Helen's paper, a good sense of quite the boundaries of it, though.
Leah, you want to?
Hi, Ruth. Thank you for the great talk. Very, very interesting. Just a philosophical question. If you were working on a model like this with an experimental group before they published, would you do anything differently? I mean, would you ask them to do something different, or would you put together your models in a different way? Yeah.
I think it's a really good question. So we're working on something at the moment that tries to automate the process of deciding when to measure, and asks kind of when are the most important time points, right? So if I could, take 10 images, when should I, over a 48-hour time window, what's the most sensible thing to do? And so we're doing that in a non-spatial context, but I think it will port over. So that's one thing I think I'd like to be able to do. And I think it's probably not necessarily what I would do before building. I mean, I think it would just be more of an iterative process, I think, really, than, okay, hey, we have this data set, what can we do? And maybe just learning kind of on the fly when is sensible to measure, or what could I do to maybe tackle this question a bit more about the different diffusion coefficients. So one of my suspicions is that we could learn more if we actually went to an individual based model, because we've got positions of every cell in this assay are clearly identified, and then there are clear clusters. I think you can go back up, it's not too annoying. There are there are clearly clusters, and sorry, it's going to share screen again. There are clearly clusters in there And so I think maybe we could learn a bit more about how quickly cells are moving by thinking about the spatial structure. So I think it's important. Yeah, I mean, I think it's important to kind of just learn, if I had an idea, learn about what can be captured and what can be measured. So, you know, fundamental question is, can you image quick enough to be able to track individual cells, right? I think that's probably the fundamental bit one for us. Why not?
Thank you.
Was there one from, I think there's another one in the
Yes.
chat from May, is that right?
Yes.
How difficult would it be to include known values of diffusion coefficients and knew that they were different? Yeah, so good question. I think the way in which I might approach this would be to think about having an informed prior distribution, so that I'd incorporate that previous knowledge into the model and then assess the extent to which that previous knowledge was consistent with the data. I think here I'm not entirely convinced that these are NDCK monolayers. I'm not entirely convinced that there's a huge amount of evidence to suggest that there is a go or grow kind of type behavior here. So it might just be that actually truly they have the same diffusion coefficient in it, or that we can't pin that down. But yeah, I mean, certainly I think if I had prior knowledge, I could incorporate it that way. But I think really the kind of probably sensible way to get a handle on this question about diffusion coefficients is maybe to track cells.
Okay, there's another question from Harsh. He says, thank you for the interesting talk. Similar to the previous question, can we say that practical identifiability depends on the set of parameters being estimated? If so, for model 1, could we use iterative methods to estimate D1, keeping D2 equal to D first pass, and then use the obtained D1 to estimate D2?
I guess, so maybe Harsh, you're asking, I guess, can you kind of iteratively almost like generate kind of and refine posteriors for each parameter, and then use that in a to maybe hope to do better here? I don't know, it's a really good question. Actually, we haven't tried that. I don't have a good sense of the answer, but it's certainly something that we could try. And my hunch is that we it wouldn't make a difference. I think that the signature is just, I think there's just not enough information there. And I should highlight that actually one of the issues, I think, to some extent here is that it's all at very low cell density. So I'm not sure. It might be that they're also, if we kind of look at things at much higher cell density, then we need to change the diffusion coefficients as well and have them depend on density. I don't know. But yeah, it's a really good question, and I think we could take a look.
Okay. Shibashi asks another question. How much does the active nature of cell movement influence the spatial dynamics?
So for sure, it's the case that if you turn off, so that there's a, you know, this is a kind of essentially a traveling wave like behavior, right? And we need the combination of both motility and proliferation to see these kinds of patterns. So one thing that is really nice actually with this model that we wrote down, and it's in the preprints, is that you can go away and you can do a traveling wave analysis, and you can look at how essentially the speed of the traveling wave depends on the model parameters. And actually, because when you do a traveling wave analysis, you linearize, it turns out that you don't care about what the carrying capacities K1 and K2 are, right? Everything just ends up depending on the diffusion coefficient and the two rate constants by memory, but it's all there. And you can, one of the nice things about that is that then you can actually ask the question, when is it important to consider that you've got these two different populations in the model, and when could you happily collapse it to a model that's just one population? And you can show that in certain given certain parameter constraints or in certain regions of parameter space, that you can in fact do that.
Thank you. Are there any other questions?
I had a question, Ruth. You showed the density profiles and that they were a little bit lower, I think, than the data. And I was wondering, it's kind of two-part. One, what you think is kind of causing that, like what is not in the model that might produce that. And the other thing I was wondering is, have you thought about other types of summary statistics that you could use and kind of collect from the data with not having to generate new data, but just like on the data that you have that you could also use for the estimation?
Yeah, really good question. So I think what happens here is quite similar to what happens with a lot of scratch assays, is that when you make the scratch or as you remove the barrier, it really upsets the cells. And it takes maybe on the order of 12 hours or more for them to sort of settle down and for them to begin proliferating and moving, so to re-enter the cell cycle, really. And so I think some of what happens is related to that actually this model maybe isn't valid kind of at very, very early times. And that I think propagates through to the parameter estimates. And we've talked long and hard in this paper and in some of the others actually about what to do about that, because it's non-trivial. I think a good question. Then I think the other point, different summary statistics. I think here we just looked at radial density profile because that seems to be a natural thing to think about. Given we were working with a PDE model, but I think if we were to work with, and this just goes back to what I was saying in response to Leah's question, I think if we were to work with an individual-based model, which would be great to do at some point, then there's a much richer set of information in here than we're actually using. So thinking about the kind of roughness of the edge, thinking about the essentially spatial patterning of cells in different phases of the cell cycle, I think could be really informative. I guess we just need to think about the extent to which it's possible to do that, to calibrate an individual-based model given a data set of this size. I think actually we haven't necessarily tried that yet, and whether we would, in moving to an individual-based model, introduce new parameters that are hard to estimate. But yeah, something I'd really like to do.
Thank you.
Thank you, Ruth. I'll say it again. Great talk. This was, and thank you all. This was a great session from all the speakers. Ander, Adam, Shibashi, and Ruth. You made it a great morning session. We really appreciate this. And thank you all for joining. So we still have a few more minutes. So if anybody has anything to discuss, it doesn't have to be just about Ruth's talk, but in anything in general, the floor is open. Or if you've had enough of us, then you can also leave. But yeah, so we have our next session tomorrow, a little early, 6 a.m. Eastern time.
7 a.m. I think.
Oh, 7 a.m. I keep saying 6 because it was the time change. Sorry, I have it in my brain, but it is a little later for the European time zone, and we have a nice set of speakers also planned. So the schedule is here in the chat. Please take a look. We look forward to seeing you again. Is there any questions, discussions? And feel free to let us know if there's anything that needs to change or improve or add. We're happy to hear. No?